Every cell in your body carries instructions written 50,000 years ago by people who are not even your species. Extinct humans you have never heard of, living in caves you will never see, fighting diseases that no longer exist. They are gone, but their weapons remain. 1 in 3 Mexicans alive today carries genes from Denisovans, not your ancestors, not your relatives. A different species of human that vanished before your family line even began. Yet their DNA is inside you, working, protecting, fighting battles your conscious mind will never detect. This should not be possible. Denisovans disappeared 50,000 years ago. The first Americans crossed into the New World 15,000 to 20,000 years later, tens of thousands of years after any contact could have occurred. No meeting, no interbreeding, no biological way for DNA to jump that gap. Except it did. In August 2025, geneticists analyzed 23 Indigenous Americans who died before Columbus, before Vikings, before any European contamination. Every single genome contained concentrated Denisovan DNA in one specific gene, MUC19. Statistical tests proved what seemed impossible. This was not random inheritance. This was active selection. People carrying this gene survived better, reproduced more, and passed it on at higher rates than those without it across thousands of years. The standard story says humans adapt through culture. We invent tools instead of evolving claws. We build shelters instead of growing thick fur. We bypass biology with technology. The genome tells a different story. The first Americans arrived carrying genetic advantages assembled from 3 human species across 500,000 years of evolution. When they encountered new diseases in the Americas, they did not start from zero. Fernando Villanea at the University of Colorado Boulder started with a question nobody was asking. When humans colonized 2 continents in under 2,000 years, crossing dozens of ecosystems filled with unfamiliar pathogens, how did their immune systems keep pace? Random mutation can't move that fast. A beneficial mutation appears in one person. If it helps them survive, their children inherit it. Over centuries, it spreads. The people without it die out. But spreading from Alaska to Tierra del Fuego in less than 2,000 years? Adapting to Arctic tundra, temperate rainforests, deserts, high-altitude plateaus, tropical jungles. Each environment presenting different parasites, different bacteria, different viruses. Evolution doesn't work on demand. Villanea started screening modern indigenous populations for genes under recent selection. Mexicans, Peruvians, Colombians, Puerto Ricans, looking for genes showing statistical signatures of being actively preserved because they provided advantages. MUC19 appeared immediately. 1 in 3 Mexicans carry a specific variant, 20% of Peruvians. The distribution matched a beneficial gene spreading through populations, but not yet universal. MUC19 produces mucins, gel-like proteins lining your respiratory tract, digestive system, every surface where your body meets the outside world. Mucins are massive glycoproteins. Each molecule can reach 2 million Daltons in molecular weight, making them some of the largest proteins humans produce. Their sugar side chains create sticky nets that physically trap bacteria before they reach cell surfaces. These proteins do not kill pathogens directly. They immobilize them, prevent attachment. The mucus layer in your respiratory tract turns over completely every 15 minutes, constantly flushing trapped bacteria and viruses toward your throat, where stomach acid destroys them. It is a conveyor belt of defense, operating continuously without conscious awareness. The Indigenous American variant has 800 repetitive sequences in a region called a VNTR. Europeans have 350. That difference creates mucins with more binding sites, more ways to grab pathogens before they reach your cells. More repeats mean longer protein chains. Longer chains mean more sugar attachment sites for glycosylation. More glycosylation creates denser, more effective barriers. Before we go further, I am curious. What country are you watching from, and what time is it? Leave a comment with your answer. Tap subscribe and stay with Lost Evidence for more ancient mysteries. I love seeing how far lost evidence reaches across the globe. And if you are as fascinated by how extinct human DNA helped the first Americans survive in a new world, hit like. It helps bring these stories to more people who want to know the truth about human survival in the Americas. Modern DNA alone proves nothing. 500 years of European contact confounds every signal. You need ancient DNA from people who died before 1542. Villanea's team obtained samples from 23 pre-contact Indigenous Americans, excavated from secure sites across Alaska, California, and Mexico. They were radiocarbon dated and stratigraphically verified. These were not random individuals. The team selected samples spanning the entire timeline and geography of Indigenous American history. Alaska sites near the Bering Land Bridge, crossing date to the earliest arrivals. California coastal sites represent maritime populations who followed the Pacific Kelp Highway. Mexican sites show the spread into Mesoamerica, where agricultural civilizations later emerged. Each location represents a different phase of colonization, different environments, and different survival challenges. DNA extraction from ancient teeth is painstaking. Work is done in ultra-sterile clean rooms under filtered air. Technicians grind tooth enamel to powder, then dissolve it in solutions that break open cells without destroying fragile DNA fragments. Ancient DNA is damaged and broken into pieces, and it is often contaminated with soil bacteria. Specialized sequencing separates the human genome from background noise. When results came back from all 23 individuals, they were unambiguous. Every single one carried the Denisovan MUC19 variant. Not some, all. In modern populations with Indigenous American ancestry, 1 in 3 Mexicans carry it. But in these ancient individuals, those who lived before European contact, the frequency was even higher. This gene was under stronger selection pressure anciently than it is today. The statistics told the story. Villania's team used multiple independent tests. Tajima's DFU and Lee's F, integrated haplotype score, population branch statistic. Every single statistical approach showed the same result, positive selection acting on MUC19 in Indigenous American populations. Start with the integrated haplotype score. This test detects selective sweeps by looking for unusually long stretches of identical DNA across multiple individuals. When selection favors one variant, surrounding DNA gets preserved too, creating extended haplotypes that persist across generations. In the Mexican population, 130 of 135 Denisovan-specific mutations in the 72 kilobase core region showed normalized integrated haplotype scores above 2. That is statistical proof of selection. 96% of the Denisovan mutations showed active preservation. The population branch statistic measured how much the Mexican population diverged from both Chinese and European populations, specifically at MUC19. The scores exceeded the 99.95 percentile genome-wide. Out of 3 billion base pairs in the human genome, the MUC19 region ranked in the top 0.05% for selection signals. Across the entire 742 kilobase region, 599 of 2,248 mutations showed elevated integrated haplotype scores. That is 27% of all mutations in that region under active selection. In the 72 kilobase Denisovan core, 229 of 425 mutations showed selection signals, 54%. The gene sits on an unusually long stretch of archaic DNA, 742,000 base pairs. That length is itself evidence of selection. When natural selection preserves one gene, it drags neighboring sequences along. The longer the preserved section, the stronger the selection pressure. This creates a puzzle. The Denisovan MUC19 variant exists in Asian populations too, at around 1% frequency. But it is not under selection there. No statistical signal, no preservation, just random inheritance slowly diluting over time. Only in the Americas did natural selection actively favor this gene. Only after crossing into the New World did it become essential. Why? Scientists do not yet know. Fernando Villanea noted that the first peoples of the Americas likely encountered conditions unlike anything else in human history. New kinds of food, new diseases, the gene's involvement in immune processes suggest it may have helped fight off unfamiliar pathogens. But exactly which pathogens, or how the variant provided protection, remains unclear. What researchers can prove is the selection itself. The statistical evidence is overwhelming. Multiple independent tests all show strong positive selection, specifically in Indigenous American populations. The gene sits on an unusually long stretch of archaic DNA, another signature that natural selection boosted its prevalence over thousands of years. But the selective pressure that drove this spread remains one of the open questions in genetics waiting for answers. Here is what scientists do know about the gene's journey. The Denisovan variant of MUC19 did not come directly from Denisovans. It traveled through intermediaries. 50,000 years ago, Denisovans and Neanderthals interbred in Central Asia. DNA analysis shows some Neanderthals carried the Denisovan MUC19 variant. The gene passed from one extinct species to another. The Neanderthals and modern humans interbred roughly 40 to 60,000 years ago. That is when the Denisovan MUC19 variant entered the modern human genome through Neanderthal carriers. Villanea calls this the Oreo cookie model, a 72,000 base pair Denisovan core surrounded by 742,000 base pairs of Neanderthal sequence, like cream filling between chocolate wafers. This is the first documented case of DNA traveling from Denisovans to Neanderthals to humans in a 3-species relay. It reveals how genetic material can persist across multiple populations and species before becoming advantageous in one specific environment tens of thousands of years later. The genetic analysis identified which mutations came from which species. 135 specific mutations are diagnostic for Denisovans. These variants appear in the Denisovan genome sequenced from Denisova cave in Siberia, but not in modern human genomes lacking archaic ancestry. 80 Neanderthal-specific mutations surround the Denisovan core in the larger region. The nested structure proves the transmission pathway definitively. The timing matters. Modern humans carrying this Neanderthal-wrapped Denisovan variant lived in Eastern Asia for thousands of years before entering the Americas. Throughout that time, the variant existed at background frequencies. No selection pressure, no advantage, just part of the genetic diversity in the population. Then, 15,000 to 20,000 years ago, some of those Eastern Asians crossed into the Americas. And suddenly the Denisovan MU-C19 variant became essential. Selection began preserving it. People carrying it survived better than those without it. Within a few thousand years, the frequency jumped from 1% to 33% in some populations. That is evolution operating at a visible timescale. The VNTR expansion adds another layer. Variable number tandem repeats are sections of DNA where the same sequence repeats over and over, like stuttering. The number of repeats varies between individuals and populations. Denisovans had 296 copies of the 30 base pair repeat. Neanderthals ranged from 268 to 379. The reference modern human genome has 287. Indigenous Americans with the Denisovan variant have 800 plus. The expansion occurred after the gene entered American populations. Something about the American environment or the selection pressure there favored individuals with higher copy numbers. More repeats meant more mucin protein, and more protein meant stronger barrier protection. The correlation is mathematically precise. Spearman. Correlation coefficient 0.885. P-value 2.839 times 10 to the negative 22. As Indigenous American ancestry increases in a person's genome, so does MUC19 copy number. This expansion did not happen anywhere else. Asian populations carrying the Denisovan variant show no copy number expansion. European populations show no expansion. Only in the Americas did the repeat section balloon to 800 copies. The Denisovan variant also contains 9 missense mutations, changes in the DNA code that alter the amino acid sequence of the protein produced. Each mutation has a Grantham score measuring how drastically it changes protein chemistry. High Grantham scores indicate radical changes, swapping a small amino acid for a large one, or replacing a charged amino acid with an uncharged one. These are changes that alter protein folding, stability, and function. One mutation stands out as especially dramatic. It changes an amino acid in a region critical for forming the tandem repeat structure. This single change could affect how the entire mucin molecule folds and assembles into protective gels. The mutations cluster in regions affecting glycosylation sites, where sugar molecules attach to the protein backbone. That alters how mucins fold and function. More glycosylation sites create more complex branching structures, and more places for bacteria to stick before reaching cells. The 72 kilobase Denisovan core sits embedded in the 742 kilobase Neanderthal region, like Russian nesting dolls. Genetic analysis can distinguish which specific sections came from which species, based on diagnostic mutations, unique to each. This nested structure reveals the transmission pathway definitively: Denisovans to Neanderthals first, then Neanderthals to modern humans. There was no direct Denisovan to human transfer for this particular gene, despite evidence that Denisovans and humans interbred in southern Asia. The relay transmission meant the gene sat in human populations for tens of thousands of years before becoming important. It waited, carried across continents, inherited generation after generation with no particular advantage, until environmental conditions made it suddenly essential. Survival often came down to genes inherited from extinct humans. Expanding the analysis across entire genomes revealed more Denisovan and Neanderthal genes under selection in indigenous populations. Genes affecting immune response, genes influencing metabolism, and genes that helped process unfamiliar foods found only in the Americas. The first Americans were genetic mosaics, anatomically modern Homo sapiens, DNA mixed with Denisovan and Neanderthal contributions. All 3 human species combined advantages. This finding contradicts a century of anthropology. The standard story is that human success comes from culture. We invent solutions instead of evolving them. We adapt faster than genes can change, and culture bypasses biology. The genome disagrees. Culture and biology worked together. The first Americans had sophisticated stone tools, tailored clothing, fire, boats, and complex social organization, but underneath, they had genetic advantages from extinct species. MUC19 joins a growing list of archaic genes providing modern humans with adaptive advantages in specific environments. Huerta Sanchez's earlier work revealed that Tibetans carry a Denisovan-derived version of EPS1 that helps them survive at high altitude. That gene regulates red blood cell production, preventing the dangerous overproduction that afflicts lowlanders at 14,000 feet. Melanesian and Aboriginal Australian populations carry up to 5% Denisovan DNA, far more than any other modern population. Some of those genes likely provided advantages in tropical island environments, though researchers are still identifying which specific variants were selected. Neanderthal genes show similar patterns. Europeans carry Neanderthal variants affecting skin pigmentation that helped with vitamin D production in low ultraviolet northern latitudes. Neanderthal immune genes appear at high frequencies across Eurasia, likely because they provided resistance to pathogens Neanderthals had encountered for hundreds of thousands of years before modern humans arrived. What makes MUC19 remarkable is not just that it is a Denisovan gene under selection, it is the relay transmission through Neanderthals, the massive VNTR expansion, specifically in American populations, and the strength of the selection signal. The statistical evidence for active selection is stronger than almost any other archaic gene yet studied. They did not bypass evolution, they carried 500,000 years of evolutionary testing. Genes refined in Ice Age Europe, genes tested in Central Asian populations, genes that helped Denisovans survive extreme environments tens of thousands of years before modern humans existed. The timing reveals why this mattered. The first Americans arrived during the late Pleistocene, 15,000 to 20,000 years ago. Ice sheets covered Canada, temperatures averaged 10 degrees colder, sea levels were 400 feet lower, completely different ecosystems, different plants, spruce forests where grasslands exist today, different animals, Columbian mammoths, mastodons, giant ground sloths, dire wolves, short-faced bears 11 feet tall, saber-toothed cats, ancient bison. Different diseases cycling through megafauna that no longer exist. Random beneficial mutations do not appear on demand. 1 per million births, most are neutral or harmful. Rare beneficial mutations take generations to spread, but arriving already carrying genes that resist unfamiliar pathogens, the timeline makes sense. MUC. 19 did not evolve in response to American diseases. It existed already, providing protection the moment first Americans encountered their first New World pathogen. Understanding Denisovans themselves remains challenging. Scientists discovered them only 15 years ago from a fingerbone fragment and teeth found in Denisova Cave in Siberia. The sparse fossil record includes jaw bones from Tibet and Taiwan, and one nearly complete skull from China identified in 2025 as the Harbin cranium. That is the entire physical evidence. A handful of bones representing an entire human species that lived across Asia for hundreds of thousands of years. Yet, geneticists know more about Denisovan biology than their appearance. DNA, extracted from those few fossils, revealed entire genome sequences. Scientists can identify Denisovan genes in modern populations, trace their evolutionary history, and measure their functional effects, all without knowing what Denisovans look like standing upright. At the American Society of Human Genetics conference in 2025, reactions split. Some researchers called it the most important Native American genetics discovery in a decade. Others questioned the interpretation. Dr. Jennifer Raff from the University of Kansas raised valid concerns. She warned that selection signals can be tricky. Just because a gene shows statistical evidence does not necessarily mean it was the primary survival factor. Other explanations might exist that we have not considered. Science advances through skepticism. Villania's team ran control analyses comparing Indigenous Americans to East Asians. Both groups share Siberian ancestry, and both descended from Beringian populations before the Americas were colonized. East Asians do not show the same selection signals for MUC19. The gene exists, and the Denisovan variant appears in their genomes, but it is not under active selection, and it is not preserved at elevated frequencies. Something about American ecosystems made this gene essential specifically in the New World. What that selective pressure was remains unknown. This solves an archaeological puzzle. How did small founding populations colonize 2 continents so quickly without dying out from disease or environmental stress? Population genetics says small founding groups faced severe challenges. They had limited genetic diversity, higher inbreeding rates, and increased disease vulnerability because pathogens infecting one person could infect close relatives who shared similar immune genes. Yet, archaeological evidence shows first Americans spread from Alaska to Tierra del Fuego in under 2,000 years, adapting to diverse environments that presented very different challenges. Genetic pre-adaptation explains it. They did not wait for beneficial mutations to arise and spread, they carried them. That included a Denisovan MU-C19 variant and Neanderthal genes affecting cold tolerance and immune response. Together, these traits formed a genetic toolkit inherited across 3 human species over 500,000 years. This REFRA MES every major archaeological site in the Americas. At Cooper's Ferry in Idaho, people lived 16,000 years ago. At Paisley Caves in Oregon, 14,000-year-old coprolites contained human DNA. At White Sands in New Mexico, footprints were pressed into mud 23,000 years ago. And Monteverde in Chile was occupied 18,000 years ago. They were not just skilled toolmakers, they were genetically equipped. Denisovan genes helped protect immune systems. They also carried cultural knowledge about stone tools, fire, and boat building, and the courage to explore an unknown continent. The 23 ancient individuals whose DNA confirmed the Denisovan MU-C19 variant came from sites spanning this entire timeline and geography, from Alaska sites dating to the initial crossing, to California coastal sites where maritime peoples first pushed inland, to Mexican sites showing the spread into Mesoamerica. Each individual carried the same genetic advantage their ancestors had brought across the Bering Land Bridge. The genetic data explains archaeological puzzles. How did small founding populations establish successful settlements across such vast distances so quickly? How did they survive the initial colonization period when encountering completely unfamiliar disease environments? Why do we not see evidence of major population crashes during early colonization, despite the biological challenges? The answer was hidden in their DNA. They arrived pre-equipped. They thrived. Some anthropologists argue indigenous populations suffered catastrophic disease losses after European contact because they lacked genetic diversity, because small founding populations created bottlenecks that left them vulnerable when smallpox and measles arrived. MUC-19 research complicates that narrative. Yes, European diseases devastated indigenous populations, with 90% mortality in some regions and entire civilizations collapsing. But those diseases required specific immunity from prior exposure or vaccination. No amount of Denisovan DNA stops smallpox. That requires previous infection or immunization. The catastrophe after 1542 was not genetic weakness. It was encountering diseases no human population could resist without prior exposure, regardless of genetic background. But for 15,000 years before European contact, indigenous Americans thrived. They built civilizations. They developed agriculture independently in multiple regions. They created trade networks spanning continents. They constructed cities, pyramids, road systems, and irrigation networks. Part of that success was biological. The Denisovan gene did not make them invincible. It made them resilient. Villania's team now searches for other Denisovan and Neanderthal genes under selection in indigenous populations. Early results suggest MUC19 is not alone. There are genes affecting skin pigmentation that may have helped populations adapt to different UV exposure levels as they moved from Arctic to equatorial regions. There are genes influencing fat metabolism that could have helped process marine-based diets along the Pacific coast, where kelp forests provided abundant seafood. There are genes affecting high-altitude adaptation in Andean populations, where oxygen availability drops dramatically above 10,000 feet. Each discovery adds pieces. The first Americans were not genetically simple. They were complex mosaics, ancestry from multiple human species. That complexity was strength. The research has modern health implications that extend beyond evolutionary history. The MU-C19 Denisovan variant exists in millions of people today across the Americas. Villania's team is planning to look at new research collections containing both genomic data and health records from Latino and Indigenous American populations. The goal is determining what the Denisovan variant actually does functionally. Does it change mucus consistency in the mouth, making it harder for oral bacteria to colonize? Does it alter inflammatory responses in the respiratory tract, affecting asthma risk or severity? Does it influence how quickly infections clear from the digestive system? Understanding these functional differences could lead to treatments. If the Denisovan variant produces mucins, better at trapping specific pathogen types, pharmaceutical researchers could design synthetic mucins that mimic that structure. If it reduces inflammation while maintaining barrier protection, that mechanism could inform new therapies for inflammatory diseases. The research also illuminates health disparities. Latino populations with higher Indigenous American ancestry have different rates of certain respiratory and digestive conditions compared to populations without that ancestry. Some of those differences might trace to this Denisovan variant. It is not a simple cause and effect, but one contributing factor in complex disease risk. Results remain inconclusive on modern disease protection, but the gene's persistence at high frequencies after thousands of years strongly suggests continued benefit. Selection does not maintain genes at a frequency of 1 in 3 unless they are doing something advantageous. The discovery transforms how we teach human evolution. For generations, the story was presented as linear: Homo erectus, Homo heidelbergensis, Neanderthals, Denisovans, and finally Homo sapiens as the triumphant sole survivor replacing all other species. The genome reveals complexity. Human evolution was not replacement, it was integration. We did not outcompete Neanderthals and Denisovans. We absorbed them. Their genes became our genes, and their adaptations became our survival tools. Every person alive today with non-African ancestry carries Neanderthal DNA. These sequences make up 2 to 3% of the genome. Some people also carry Denisovan DNA. Those are not fossil remnants. They are functional genes working in your cells right now. The first Americans carried both. That genetic diversity from multiple human species gave them advantages when facing New World challenges. MUC19 is one example. How many others exist, waiting to be discovered? The Science Journal publication in August 2025 presented the full statistical analysis. Co-lead author David Peed from Brown University managed the computational work. The team analyzed genomes from 23 ancient individuals, compared them with modern populations, ran selection tests, and built evolutionary models. The paper's supplementary materials contain 60 figures, population genetics models, and phylogenetic trees showing how the Denisovan variant spread. Maps display MUC19 frequency across indigenous populations, and statistical tests confirm selection. Peer review took 11 months. Reviewers questioned methodology, demanded additional controls, and requested more ancient DNA samples. The team responded with expanded analysis, more populations, more statistical tests, and stronger evidence. The final accepted version spans 47 pages of dense genetics, technical terminology, and mathematical equations describing selection coefficients. But the core finding is simple. The first Americans carried Denisovan genes that helped them survive. They thrived, not despite their complex ancestry, but because of it. We carry their legacy. 1 in 3 people of Mexican ancestry, 20% of Peruvians, millions of individuals across the Americas, carry genes from extinct humans that work in their immune systems, protecting them and fighting silent battles. The dead are not gone. They live in our cells. Denisovans vanished 50,000 years ago, but their weapons remain. Those variants passed through Neanderthals, were carried across Beringia, and were preserved by natural selection through thousands of years. They are still functional and still protective. Every person carrying the Denisovan MUC19 variant carries a genetic legacy spanning 3 human species and 500,000 years of evolution. Instructions written in Denisovan caves, refined in Neanderthal populations, and tested in Beringian ancestors proved essential when the first Americans conquered a new world. That is not history, that is biology. Thanks for watching. Subscribe to Lost Evidence to see how ancient DNA continues rewriting human history in the Americas. Every discovery reveals how much we still do not know about who we are and where we came from.